Assalamualaikum and hello Dr. Nolaila. My name is Zuriani binti Ilyas and I'm from Batu Gajah Perak. I'm in year 2 as a biology student. I'm working as a dental nurse at Trono Dental Clinic, and now my task is to give you a simple but detailed explanation about C3, C4 and camp plans through my poster. So let's go. A normal plant, one that doesn't have photosynthetic adaptations to reduce photorespiration, is called a C3 plant. The first step of the Calvin cycle is the fixation of carbon dioxide by rubisco, and plants that use only the standard mechanism of carbon fixation are called C3 plants. For the 3-carbon compound 3-PGA, the reaction produced. About 85% of the plant species on the planet are C3 plants, including rice, wheat, soybeans, and all trees. In C4 plants, the light-dependent reactions and the Calvin cycle are physically separated, with the light-dependent reactions occurring in the mesophyll cells and the Calvin cycle occurring in special cells around the leaf veins. These cells are called bundle sheath cells. To see how these divisions helps, let's look at an example of C4 photosynthesis in action. First, atmospheric CO2 is fixed in the mesophyll cells to form a simple 4-carbon organic acid, oxaloacetate. This step is carried out by a non-rubisco enzyme, PEP carboxylase, that has no tendency to bind O2. Oxaloacetate is then converted to a similar molecule, malate, that can be transported into the bundle sheath cells. Inside the bundle sheaths, malate breaks down, releasing a molecule of CO2. The CO2 is then fixed by rubisco and made into sugars via the Calvin cycle, exactly as in C3 photosynthesis. This process isn't without its energetic price. ATP must be expended to return the 3-carbon ferrine molecule from the bundle sheath cells and get it ready to pick up another molecule of atmospheric CO2. However, because the mesophyll cells constantly pump CO2 into neighboring bundle sheath cells in the form of malate, there's always a high concentration of CO2 relative to O2 right around rubisco. This strategy minimizes photorespiration. The C4 pathway is used in about 3% of all vascular plants. Some examples are crabgrass, sugarcane, and corn. C4 plants are common in habitats that are hot but are less abundant in areas that are cooler. In hot conditions, the benefit of reduced photorespiration likely exceed the ATP cost of moving CO2 from the mesophyll cell to the bundle sheath cell. Some plants that are adapted to dry environments, such as cacti and pineapples, use the crassulaceous acid metabolism CAM pathway to minimize photorespiration. This name comes from the family of plants, the Crassulaceae, in which scientists first discovered the pathway. Instead of separating the light-dependent reaction and the use of CO2 in the Calvin cycle in space, CAM plants separate these processes in time. At night, CAM plants open their stomata, allowing CO2 to diffuse into the leaves. This CO2 is fixed into oxaloacetate by PEP carboxylase, the same step used by C4 plants, then converted to malate or another type of organic acid. The organic acid is stored inside vacuoles until the next day. In the daylight, the CAM plants do not open their stomata, but they can still photosynthesize. That's because the organic acids are transported out of the vacuole and broken down to release CO2, which enters the Calvin cycle. This control release maintains a high concentration of CO2 around rubisco. The CAM pathway requires ATP at multiple steps, so like C4 photosynthesis, It is not an energetic freebie. However, plant species that use chem photosynthesis not only avoid photorespiration but are also very water efficient. Their stomata only open at night when humidity tends to be higher and temperatures are cooler, both factors that reduce water loss from leaves. Chem plants are typically dominant in very hot, dry areas like deserts. C3, C4, and chem plants all use the Calvin cycle to make sugars from CO2. These pathways for fixing CO2 have different advantages and disadvantages and make plants suited for different habitats. The C3 mechanism works well in cool environments, while C4 and chem plants are adapted to hot, dry areas. Both the C4 and chem pathways have evolved independently over 2,000 times, which suggests they may give plant species in hot climates a significant evolutionary advantage.